Okay. All right. Well, thanks everybody for coming out, who's here, and also all those virtually who's attending this talk. So this is our last E&E seminar of the year, and it's my privilege to introduce Fonti Carr. And this is a this is a special talk for me because Fonti and I, this works okay, have actually been working together for an enormous amount of time. I think it's been like 7 or 8 years or something. And for those who are unaware, you might have heard me refer to Fonti as Master Carr. I do so because Fonti started off working with Martin Whiting and I at Macquarie University, where she did her master's. And she worked on these beautiful brown charismatic skinks, water skinks. And she did some beautiful work on contest competition, social learning whilst at Macquarie. But for those who know Fonti, she has an undergraduate degree with quite a lot of statistics, and she was quite keen on implementing and pursuing some more statistics for her PhD. And that led her to the Ideal Group and joining Shinichi Nakagawa and I at UNSW and undertaking her PhD there. And this is just a bunch of photos from various events that we've had over the years, Fonti. Now, Fonti's thesis is really beautiful because it combines empirical work and kind of comparative work all in one sort of nice little package. And she also was able to apply some fairly new statistical tools to answer some interesting questions about phenotypic plasticity. So Fonti's PhD has been what I think is great because it combines all of her skills and passions in one place. She loves to catch lizards, because let's be honest, who wouldn't love catching lizards? It's like the best thing ever. And she's extremely good at it. She's caught thousands of lizards, it seems, over the years. And she's also been able to pull off some extremely impressive experiments, which have dragged on for, well, somewhat ludicrous experiments, actually, Fonti, but they're amazing. But I didn't know we would be able to pull it off, but Fonti did it, and that's because she's extremely motivated, extremely determined, and amazingly organized. And she's got some absolutely beautiful data from those experiments. Now, as with all huge data sets comes quite a lot of programming and statistics and everything to try to deal with the complexities of big data. And over the years, Fonti has really developed her programming skills, and I'm just going to feature some really beautiful examples. My two favorite examples that she's produced for me over the years. And I think this is going to serve her extremely well in her next phase of her career, where she's going to be postdocing with Dan Foster and Will Cornwell, really developing her programming skills even further in terms of building R packages. And there's a little Elena, I think 6 weeks old. Now, for those who haven't had the privilege of listening to any of Fonti's talks, she's one of the most outstanding communicators you can imagine. I think for all the conferences that she's been to, I can't even remember one where she hasn't had a runner-up or an award or something, or honorable mention. It's just a constant barrage of awards for Fonti. And they're well-deserved. Okay, so Fonti's not just a workaholic. She loves to play as well, and she loves snorkeling and bushwalking, and probably most important for us, she loves to bake and cook, and we've reaped the benefits of that over the years, where she's brought some really scrumptious food to lab meetings, which makes scientific discussions really pleasant and even more enjoyable. So I'm going to quickly end here and just say probably one of the most aspiring things about Fonti is her compassion and willingness to help others. She's very altruistic. I don't know any situation where I've asked Fonti if she could help and she said no. She's always there to help, whether it be for a coding problem or to look after Lena for 10 or 15 minutes while Yen and I take a bit of a breather, or anything. She's just there to help all the time. And I know maybe, everybody here, both virtually and in the room, has probably received some of that help. So thank you, Fonti, for that passion and enthusiasm over the years. And without any further ado, I'm going to hand this over to you and hope this transition works okay. And we will have a little, I've booked out the Badger & Co. For those who might want to join for some drinks after the talk. But I'll hand over to you now, Fonti, and hope this works. Okay, stop sharing there.
Okay, how do I get? Okay. Thank you, Dan. That was it's always so surreal when your supervisor says such really nice things about you. But yeah, that was so lovely. I didn't expect that. Before I begin my talk, I would like to acknowledge the people of the Ngunnawal country, the traditional custodians of the land we meet today. I'd like to pay my respects to the elders past, present, and emerging, and extend this respect to all Aboriginal people and Torres Strait Island people who may be joining us today. So I want to preface that I'm not technically an ANU student. I started, as Dan said, I started my PhD at UNSW, and I moved here about a year ago to work more closely with Dan and to wrap up my thesis away from the big smoke. And it's been a really wonderful experience, despite bushfires, hailstorms, and a global pandemic. But I just want to thank you all for being here today, online and in person. It's going to be, yeah, it's really nice to be able to share my work with you guys.
Fonti, this is God speaking. Can you share your screen?
Oh no! Am I not sharing? Okay, no, we can do this. Oh yeah, I'm not sharing. Where's my mouse? Here we go.
There you go.
Boom. How's that, Mike?
God is happy. Continue.
Okay, so let's begin. I want to start this talk by imagining what life would be like as a developing embryo. I think life would be pretty damn uncertain, especially if you're encased in an egg, because you have pretty limited control of where you find yourself in the environment. And that's pretty troubling because a little embryo has to face whatever the world throws at it. You can't simply just pick yourself up and move to a nicer location. So over millions of years of evolutionary tweaking, embryos have developed the ability to adjust their phenotype in response to these conditions they experience early in life. This phenomenon is called the developmental plasticity. And here are some of my favorite examples in nature. In dwarf spiders, whether an adult uses these ballooning threads of silk to disperse over long distances depend on the thermal conditions juvenile spiders experience. In common frogs, the timing of when tadpoles metamorphose depends on how fast the water levels of the pond decreases. And in common starlings, if you're the runt of the family, you actually become more competent at flying and better at escaping predators, and compared to your larger offspring. And finally, in the water flea, if juveniles detect predator cues in the water column, they develop these spiky, jousty helmets as a way to defend themselves in the future. So what I want to illustrate is that developmental plasticity is incredibly widespread and plays a pivotal role for animals in coping under changing environments. And it's said that developmental plasticity evolved under situations where early life environments can reliably predict the conditions adults have to experience later in life. But maladaptive mismatches can occur when the environment changes so much so that these cues can no longer provide reliable intel. So this had led many, including myself, to wonder, well, what are the traits that are changed by these early life environments? Are they persistent through life? Are they flexible to fine tuning? And more importantly, can we predict these phenotypic changes and what are the evolutionary consequences of them? So one way to kind of get at these broad questions is to look at the effects of early life environments on metabolism. And that's simply because life on Earth requires energy. Animals have a finite amount of resources to allocate to competing demands. And that includes reproduction, growth, somatic maintenance, and survival tactics. And metabolism, which represents the idling cost of living, is said to mediate trade-offs between these competing demands, and which can shape phenotypic diversity in nature. So for a long time, people have observed that metabolism co-varies with life history in particular patterns. Fast metabolism, represented in red, is shown to be associated with fast growth, early maturity, having many cheap babies, and short lifespan. Whereas slow metabolism is associated with slow growth, late maturity, having few but really high quality babies, and longer lifespans. And this is called the slow-fast continuum, or the pace of life. And we can apply this framework in a way to understand how early life environments shape phenotypic development. And the idea is if early life environment can change metabolic rate in a persistent way, we might expect these effects to cascade onto life history and traits like growth, maturity, and lifespan. So what we currently know is that early life environments can shape metabolism. So elevated levels of parasitic load, increased food density, and the relative size of other individuals at early stages in life can increase metabolic rate. But what we don't know is whether this can then go on to affect life history. But something that's really important that we need to consider is that metabolism is highly plastic. And what I mean is that an individual's energy expenditure is dependent on its surrounding environments. And this change, in this example, this is metabolic rate, and it shows a positive relationship with acute temperature. And this is often the case in many ectotherms. This change in metabolic rate can be represented by a reaction norm. And this is defined by two quantities, the intercept and the slope. And the steepness of the slope describes how sensitive metabolic rate changes in response to temperature. And there's some really interesting work by Frank C. Bakers and colleagues that show that early life environments can also change the shape of these reaction norms. So they show that fish born in spring conditions tend to have much steeper reaction norms compared to fish born in summer. And this means that fish in spring are more sensitive to changes in the thermal environment. So I think understanding how early life environment changes the shape of these reaction norms will be increasingly more important for species that have long reproductive seasons that occupy variable habitats. While plasticity is a very effective and short-term solution, in some cases, populations may need to evolve genetic adaptations to persist in the long term. So there are two basic ingredients that you need for something to evolve. The first is the presence of consistent phenotypic variation. What I mean is that differences between individuals need to be maintained over time. So if I sample the same group of individuals at different time points, the variation among them is stable. And this is because this is the raw material that selection works with. And often this is represented as repeatability, which is a ratio of individual variation over the total phenotypic variance. The second thing you need is that some of that phenotypic variation needs to have a heritable basis, because this provides the mechanism for traits to be passed on to offspring and to subsequent generations. And this is typically represented as heritability, which is the ratio of additive genetic variance over total phenotypic variance. What complicates things is that we know that early life environment also affects these two quantities. So, for example, development under urban environments, nutritional stress, has been shown to increase repeatability of metabolic rate and behavioral traits. Whereas for heritability, development in different seasons and at different temperatures have been shown to also change this to change heritability. What these studies show is that the amount of phenotypic substrate that's available for selection to work with and the rate of evolutionary change is dependent on these early life environments. So today, the first two studies I want to share with you guys is some work that I did in my PhD that looks at the effects of developmental temperature on metabolism and life histories. So specifically, I was interested in understanding how developmental temperature affects reaction norms and the repeatability and heritability of these traits. So for these two studies, I got to work with this cute little skink, the delicate skink. They have this incredibly widespread distribution along the southeast coast of Australia. And it's also the first ever lizard to become, from Australia to become invasive overseas. Females have about a clutch size varying from 1 egg to up to 8 eggs. And I use this highly specialized technique to bring them back from the wild. And it's called mealworm fishing. And it's exactly as its name implies. You have a mealworm tied to an end of a bit of a sinker, which is tied to some fishing line and then a pole. And with some practice and patience, you end up catching quite a few on a good day. So we brought adults back from the wild and they were able to breed in our lab, and females were able to produce eggs that are used for my experiments. So I used a split clutch design where I took eggs from the same female and split them across the 2 developmental temperatures. So these developmental temperatures represent natural thermal extremes that eggs experience in Sydney populations. The cold treatment had an average temperature of 23 degrees. The hot treatment had an average temperature of 29 degrees. And both treatments fluctuated plus or minus 3 degrees to mimic naturalistic fluctuations that wild populations would experience. And so that was a time lapse of a little lizard hatching. And once lizards hatched from these treatments, they were kept at the same conditions in the lab. So this first study I want to talk about looks at specifically the effects of developmental temperature on metabolism and its thermal reaction norm. So what I did was I used closed system respirometry to measure metabolic rate. And this is when you seal a little lizard in a chamber for a period of time during which it's respiring. So you can measure the change in CO2 or O2 to measure metabolic rate. And in my study, I measured the rate of CO2 production as metabolic rate. So for each lizard, I measured metabolic rate across these 6 acute temperatures that you can see on the bottom there, 10 times in order to estimate these reaction norms for the cold and hot lizards. And this was over the course of 3.5 months. And you might be thinking, why on earth would you measure something 10 times if it's the same thing? And that's the level of commitment and data, and that's the kind of data you need to estimate the repeatability of plasticity, which is represented by the slope. And so I wanted to compare where the treatments differ in the repeatability of the slope, as well as the repeatability of the intercept, which represents the average metabolic rate. So this is what I found. Overall, thermal reaction norms were robust to changes in developmental temperature. Hot and cold lizards both share the same intercept and same slope. What this means is that lizards from both treatments respond to temperature in the same way. So before I get into my repeatability results, I want to show you what high repeatability and plasticity looks like. So what you expect for high repeatability is that there are lots of individual differences in these slopes, so the angles of the line, which is represented on the right. Whereas on the left, every line has the same angle. So in that situation, there are no differences in the slope. But this high individual variation needs to be also maintained over time in order for it to be repeatable. So what did I find? Overall, thermal reaction norms were significantly repeatable, which means that individuals respond to temperature differently and in a consistent manner. However, repeatability was not dependent on our developmental temperatures. But what about the intercepts? So in blue are the cold lizards and in red are the hot lizards. You can see across all the acute temperatures, metabolic rate, the repeatability of metabolic rate was lower. for hot lizards. And that means that hot lizards, their metabolic rate is less variable compared to cold lizards. So in summary, there was no impact of developmental temperatures on thermal reaction norms. However, development under hot temperatures reduce the repeatability of your metabolic rate. And this is a bit worrying because temperatures are expected to increase with global warming. However, it looks like delicate skinks harbor sufficient consistent variation in the slope, which may be enough for selection to work with and may enable populations to evolve suitable responses in future climate. Okay. For this next study, I looked at the effects of developmental temperature on growth. And the idea is that growth slows down under cold temperatures and takes place over much longer periods of time, whereas growth accelerates under warmer temperatures and takes place over a shorter period of time. And this temperature variation in growth has been used to describe and explain broad-scale ecological patterns in body size. So the temperature size rule describes situations where species that occupy in cold habitats tend to reach much larger body size compared to the same species found in warm habitats. So typically growth is quantified by taking measurements of body mass at a few time points, and the change in body mass is equated to growth. But we know that growth doesn't scale linearly and it doesn't continue on forever. In fact, it actually gradually slows down and eventually plateaus. So by taking only measurements at only a few time points, what that does is that it can mislead estimates of growth rate, and you can also miss out interesting patterns like catch-up growth. And so in order for us to understand variation in growth, what we need is to quantify these growth curves in greater detail. So I want to quickly pivot and talk about what contributes to variation in body mass. So variation in body mass is consisted of several components. Some of that variation is due to genetic differences between individuals. Some of it is because of maternal effects. So attributes of your mum can affect your body mass. So think about like maternal care or nutritional provisioning, which can affect body mass. Some of that remaining variation is due to the environment. So this can include abiotic factors like resource availability, as well as social aspects of the environment, like who you're hanging out with. We can further classify this environmental variance into permanent environmental effects. So these are environmental factors that has a persistent effect on an individual's body size. And so early life environments would be technically considered as a permanent environmental effect. So remember how I said heritability is a ratio of additive genetic variance over total phenotypic variance. Well, this is just that total phenotypic variance broken down in its main constituents. And heritability is said to, is often referred to as the evolutionary potential of a trait, because if selection acts on a trait that has higher heritability or a large genetic component, it's said to evolve a lot faster compared to a trait with a low heritability. What's really interesting is that just like mass that changes with age, these genetic and non-genetic sources of variation also varies across ontogeny. So here is just some examples of them. It's widely documented in mammalian systems. So here in the middle, bighorn sheep, you can see that additive genetic variance increases over time. And that is also reflected here in the heritability. So for this study, I wanted to look at how developmental temperature shapes these growth curves and these ontogenetic patterns in genetic and non-genetic variants. So for this study, we used a half-zip design where a single male was mated with multiple females, and the eggs from the females were incubated in the same way as I described before. And once babies hatched, we took a small DNA sample for SNP analyses, and used that. And with the help of Scott, we estimated a genomic related matrix, which describes the proportion of your genome that is shared by that's shared within other individuals. And with the help of some amazing interns, we measured body mass for over 200 individuals over 2 reproductive seasons. And on average, a lizard had about 16 measurements for just over a year. And using an animal model, we were able to partition out these genetic and non-genetic sources of variation and see how they changed with ontogeny. So here is what I found. Overall, growth was not influenced by the effects of developmental temperature, but this wasn't really surprising because we didn't see an effect on metabolic rate. However, cold lizards were on average 9% heavier at hatching, and this difference in mass was persistent through life. Overall, the genetic and non-genetic sources of variation changed with age. Additive genetic variants increased with age, and we think this has something to do with the genotypic changes associated with sexual maturity, which is about 420 days in this species. Maternal effects initially declined, and this is what I expected because mums can no longer really interact with their babies and to affect their body mass. But we also detected this resurgence in maternal effects, and this was unanticipated, but it's actually been described in Japanese quails with tarsus length. And these results sort of imply that there are other aspects of mums that can influence offspring body mass at a later stage in life. Cold lizards, when they hatched out, had more environmental variance compared to hot lizards. And this, I think this had something to do with the rate of development at these different incubation temperatures. So let me walk you through this. The rate at which an embryo develops increases at an exponential rate with temperature. So what I think is happening is that hot lizards may be more constrained in their developmental rate, and as a consequence, have to hatch out early. And this is a 40% difference in incubation duration. And you can see there's less variation in incubation, suggesting that they are a bit more constrained. So under cold temperatures, the embryos had a lot more time to develop. And not only that, they were able to, they might be able to maximize all the yolk resources in the egg. And this is known to vary across females and clutches. And that may be why cold lizards started life with a bit more environmental variance compared to hot lizards. So when you put these different components together, overall, we didn't find differences in heritability. However, similar to additive genetic variants, it also increased with age. So in summary, there was no effect of developmental temperature and growth, but it did affect the initial starting mass of cold lizards, and this was persistent through life. While there was no effect on heritability, these dynamic ontogenetic patterns in genetic and non-genetic variants implies that the evolutionary potential body size may be greatest at particular life stages. Okay. And more to go. Okay, so my first two studies looked at metabolism and life history, or growth rate specifically, independently. For this last study, I want to try to bring these two elements together. So life history strategies on Earth in the animal kingdom is incredibly diverse. For example, hummingbirds mature in 3 months and lives a short life of 3 years, whereas African elephants takes 9 years to reach sexual maturity and lives a healthy long life of 65 years. And this pattern still remains when you control for differences in body size. So why are there such big disparities in life history strategies still remain to be a really baffling mystery in nature. So how animals allocate energy to competing processes like reproduction, growth, and somatic maintenance is said to shape life history strategies. So for example, if a species invests more in cheap reproduction and rapid growth, they have less energy for maintenance. And as a consequence, they mature early and have shorter lifespans. Whereas a species that invests more heavily into maintenance and having high quality offspring will tend to grow more slowly, mature later, and have longer lifespans. However, there are a number of factors that can affect energy acquisition and allocation. And so, for example, metabolic rate is said to shape life history variation because it determines how large an animal's energy budget is. This is called the rate of living hypothesis, and species with high metabolic rates or high idling cost of living is said to have a fast pace of life compared to species with a slow metabolism. However, some people think it's all about reproduction and how much energy puts into reproduction. So species that invest more heavily into reproduction have less energy for these other processes, and as a consequence, have a faster pace of life. And this is in contrast to species that invest a lot less in reproduction, who then have slower paces of life. But we also know that many aspects of the environment can affect energy acquisition and allocation and can therefore shape these life history strategies. So for example, at high population densities, competition is expected to increase, so acquisition for resources is expected to be constrained. So this can affect, so species tend to invest more in rapid growth as a way to outcompete one another, and this would lead to faster paces of life. But this environmental constraints hypothesis is really difficult to test because at an interspecific level, because it's quite hard to get species data for some of these variables. But one way to get around this, one way to test this hypothesis is to look at how environment constrains activity seasons. So the activity constraints hypothesis predicts that under favorable environments, species that permits longer activity seasons, species are able to acquire more resources, which they are able, and they have more opportunities to reproduce and to grow. And this is expected to lead to a fast pace of life. Whereas species that occupy in unfavorable environments, where activity is constrained to very specific and certain times of the year, species will then have less opportunities to forage and to grow and to reproduce, and as a consequence, will have a slower pace of life. So these are the four main hypotheses that are used to explain life history variation. And I wanted to test which one is the most important in explaining variation in life history in a very special group of animals. So welcome to the weird and wonderful world of reptiles and snakes. So lizards and snakes. So lizards and snakes are a really wonderful system because they exhibit two modes of a variation in modes of reproduction. So unlike birds that strictly lay eggs and mammals that only give birth, lizards and snakes can do both. So this provided me with a neat opportunity to look at how differential investment in reproduction between egg laying and live birth can influence life history. So generally speaking, live birth is considered a more costly reproductive strategy because mums have to retain developing embryos until gestation is complete. And so she's carrying around a lot of extra weight, and which puts her at increased risk of predators. And she also has to actively bask in order for her embryos to develop. So under these circumstances, I predicted that live birth is associated with high investment in reproduction and therefore have potentially faster paces of life. So in my study, I calculated reproductive investment as the number of offspring multiplied by offspring mass divided by mother's mass. And you can think of this as how much mass mum puts into her average offspring. To test this question of what predicts pace of life, I collected species data for over 500 lizards and snake species for age and maturity and lifespan. I used a bivariate mix model to test the partial effects of all those hypotheses that I introduced, and I included metabolic rate, reproductive investment, the length of each species activity season, and latitude. And latitude here represents the environmental constraints hypothesis. And that's because lots of factors like temperature and rainfall that are expected to affect energy acquisition and allocation are correlated with latitudes. We felt this would be a good surrogate for this hypothesis. So I also included body mass and research effort in this model because we know that larger animals tend to live longer and well-studied species tend to be, have more accurate estimates of life history traits. So to account for phylogenetic relatedness of these different species, I also included a molecular phylogeny. And here you can see the distribution of egg layers versus live bearers across the phylogeny. So this is what the data looks like. On the x-axis, got age at maturity, and on the y-axis, I've got lifespan. Egg layers are represented by these yellow circles. Live bearers are by these green triangles. So overall, you can see that while live bearers mature at a later age, they don't tend to live a lot longer compared to egg-laying species. This little weird group up in the corner are your New Zealand geckos, and they're known to have a very slow pace of life because they essentially live in a predator-free island. So overall, both these traits exhibit quite high phylogenetic signals. What that means is that closely related species tend to be more similar compared to more distantly related species. But more importantly, what predicts pace of life? So here I've got the 4 key hypotheses that I wanted to test on the left-hand side. In the middle, I've got age at maturity, the right lifespan. This dashed line represents no effect of any of these hypotheses. And again, yellow is egg layers, green live bearers, and black is an overall estimate for both reproductive modes combined. So points on the left-hand side of this line are associated with lower trait values and lower, so what I mean is low, early maturity and short lifespans. So traits that are associated with fast paces of life. Points on the left-hand side is associated with higher trait values, so late maturity and long lifespans, so traits that you're expected to be related to slow paces of life. So I wanted to ask, what was really surprising was that we didn't find any differences in any of these effects between egg-laying species and live-bearing species. So for the purpose of this talk, I'm just going to focus on the overall estimate for lizards and snakes. So across our data set, we found no support for the rate of living hypothesis, but it's important to know that the effect size is in the direction that's predicted by the rate of living hypothesis. So high values of metabolism is associated with early maturity and long lifespan. There was some support for the reproductive allocation hypothesis in only age maturity. So the more you invest into your babies, the earlier you tend to mature. Similarly, there was some support for the activity constraints hypothesis. Species that have longer activity seasons tend to mature early compared to species of short seasons. And finally, for the we found support for the environmental constraint hypothesis for both age and maturity and lifespan. And what this means, what these points means is that closer to the poles, species tend to mature later and live longer. And other studies have shown that in temperate zones, predation risk, population density, and juvenile mortality decreases. So these are all the conditions, environmental conditions, where you would expect slower paces of life to be favored. So in summary, the effects of metabolic rate, reproductive investment, and environmental constraints were the same for both egg-laying species and live-bearing species. Metabolism was not the sole driver of life history variation in lizards and snakes. And instead, my results suggest that it's the complex interplay between the environment and how animals acquire and allocate energy may be more important in driving life history variation. Although latitude is a very coarse correlate of the environmental factors, I hope this prompts future studies to look at more specific environmental variables. Okay, we made it. So some broad take homes from some of the studies I talked about today. While metabolism is reversibly plastic post-development, it didn't respond to the changes in early life environments. And similarly, this didn't lead to concordant changes in growth. However, early thermal environments had a subtle and long-lasting effect on hatching mass, as well as the repeatability of metabolic rate. While the average phenotypes and reaction norms didn't differ between these developmental treatments, there were changes in phenotypic variants and genetic variants. And this has biological importance because these are the things that determine how much phenotypic substrate is available for selection to work with, as well as the evolutionary potential of these traits. And finally, unifying theories like the pace of life is, well, it's kind of helpful to have them to predict patterns of phenotypic variation. Metabolism being the key to everything seems a little bit too sweet and too simple. And I think it's more of an exception than the rule. So now I want to spend the remainder of my talk to thank several people that were really influenced during the journey of a PhD. I think doing a PhD is kind of like playing a game of snakes and ladders. There are triumphs, there are wins, but there are also a lot of unexpected obstacles. So I just want to thank the people that really helped me keep me afloat over the past couple of years. So first I want to thank Shinichi. Shinichi, if you've ever met him, he's one of the smartest people I've ever known. And it's really nice to have such a knowledgeable supervisor that's really so willing to impart knowledge with you. So I've learned a lot throughout my PhD and feel like I've only just scraped the surface of Shinichi's vast expertise. Shinichi also has this contagious passion for science, and this was really motivating when I lost sight of the relevance of my work. I'm grateful to have a supervisor that shares the same level of enthusiasm for baked goods, as well as solving coding mysteries at work. But unfortunately, I don't share the same love for comfortable, breathable footwear. So next, I've got some nice things to say about Dan. So I've known Dan for about 6 years now. It seems like yesterday. Seems like yesterday that I was just Skyping this random postdoc to talk about potential master's projects. And today I'm still very grateful to still be working with Dan, and I hope it continues into the future. If I had to pick, I think Dan's motto would be go hard or go home. And I think this can be applied in various contexts. If you've ever been to a conference with Dan, I think you know exactly what I mean. But no, on a more serious note, I think Dan just really enjoys solving difficult problems because these are the problems that you learn the most from. And he's really encouraged me to do the same throughout my PhD. So for example, recently I asked him to help me practice for a job interview. And honestly, the practice was so much harder than the actual interview. And I think that was the point. He wanted me to prepare for the worst. So Dan is very generous with his time and his knowledge. He's in there in the battlefield with you, whether that's like catching lizards all day in Sydney heat or spending, the whole night doing science in the lab, he'll always show up for you, even if you're too proud to ask for his help. So for that and many things, I'm very grateful. Thank you, Dan. Next, I'd like to thank my family, especially mom and dad, even though they don't quite understand why I spent 6 years studying lizards. They stood by me and provided, endless support and encouragement. And that's what anyone could ever ask for. I'd also like to thank Rose for introducing me to her family, her mum, Susan. So Susan is pretty much my unofficial Australian mum. She's literally put a roof over my head while I'm here in Canberra. And that really made life here feel like home. And that was just really wonderful for the last year of my PhD. So thank you, guys. So next, I want to thank the members of Lizard Lab and all the students that helped me on my PhD there. So just without their help and their facilities, I honestly wouldn't have been able to do any of the experiments. So I'm really grateful for them being so accommodating with me, because those lizards took up a lot of space. And finally, I just want to thank my friends all over the world, especially Julia and James in South Africa, Amy and Chris in Sydney, and Sil, my soul sister in Perth. They've always provided a shoulder to lean on if I needed it, and always saw the best in me, even when I didn't. So for that, I'm very grateful. To my friends in Sydney at UNSW, thank you for reminding me that life is so much more than a PhD, and that made PhDs a lot less daunting and isolating when you know that your office mates are going exactly through the same struggles as you are. And last but not least, to everyone here at ANU, thank you so much for being so welcoming and friendly and have taken me under your wing when I first arrived. It really helped me settle in really quickly and helped me embrace Canberra life, which is filled with wonderful picnics, frisbee, biking around. It's just lovely here. So you guys have made it really difficult for me to move back to Sydney. And with all of that, I'll be happy to take some questions. Thank you.
Fantastic. Thanks, Fonti. That was great.
What was that mumble?
Can you hear me, Fonti?
I can hear you loud and clear.
Okay, great. So just to let the people online know, if they would like to ask any questions, if they just raise their hand, I can then turn on their microphone and they can ask you a question. Otherwise, if you want to take some questions in the room, but just remember to repeat the question, because we can't pick up the person asking the question on the microphone.
Sounds good.
All right.
Questions. It's always so awkward waiting for questions. Megan.
So it's not the resurgence in the maternal effects. What's going on there? Like any ideas?
So Megan asked what might be happening with the resurgence of maternal effects in the growth study. I think it has something to do with maternal genetic effects. So something to do with maybe, because mitochondria, which is really important for metabolism and growth, is passed on strictly by mothers. So maybe that has something to do with it. Yeah. Yes.
Do it affect daughters differently than sons?
So Rob just asked if maternal effects, or just in general, or just specifically maternal?
Generally, you could imagine that sort of curve, maybe if there are maternal effects that appear later on, say in life when you start to...
Yeah, okay. Let me repeat that question. So Rob just asked whether those maternal effects and body size affected lizards of different sex differently. So because lizards were quite difficult to sex at that age, they're just so tiny, they're like the length of my thumb, if not length. So we didn't account for sex in those in that study, but I think it would be, we're looking at sex-linked loci at the moment. So then maybe if we include that model and plot it by sex, we can see something more interesting. Yeah. Any questions online?
Someone's typed in a question, so I'll just read it out. This is from
Okay.
Jeff Hughes. Was there a particular reason you selected delicata for this study, or was it simply a matter of them being common and easy to catch?
Thanks, Jeff. Thanks for tuning in. That's exactly, well, delicate skinks. So there's a couple of reasons why they're really cool. So they are really common and they're really easy to keep in the lab. I mean, those are real bonuses, but mothers also have this really long kind of variation in lay date. So I think that was really interesting because that long variation in lay date spans lots of different thermal environments. We expected that, so we expected thermal environments to play an important role in the system. So that sort of was one of the reasons why. Thanks, Jeff. I think Pada had a question.
Yeah. Well, I have two, but I'll ask you one. When you were doing the metabolism trials at different time stages, the slope was going up, and when you put them in the chamber, did you start from the same temperature? As in, the lizard was all kept at 20 Celsius, and then you put them in the chamber at different temperature. So I was wondering, do you think if there's any effect on the rate of change in the metabolism as well?
So Pada asked me a long question that was hard to repeat online. Something to do with the rate of change in metabolic rate in response to temperature. I think I can answer it. So for that experiment, we randomized the order of what animals were exposed to. So there wasn't like an increasing effect of like they didn't ramp up at higher temperatures. I don't know if that answers your question. Okay.
So you only did one per day?
So we did 2 temperatures a day, and they were in a random order. So sometimes it could be 32, it could be, and then back to 24, and sometimes it could be 26 and then to 28. But because we did so many measurements and we also accounted for the order of the temperatures, we think this result is pretty robust. Cool, thanks for that question.
We have a question online. I think this might be coming to you all the way from South Africa. We'll see. So this is Julia and James.
Hi, Fonti, it's Julia. I was wondering, after when you start doing all this amazing work, there's always a lot of questions that seems to be more questions than answers that you get out of a PhD. And especially your amazing work on your meta-analysis, sometimes you can really key into certain trends. So I was just wondering, from your expertise on these topics, where do you see the future of this field moving forward, or are there particular study groups that seem to be really exciting, and what could those topics or study groups be?
Dude, that's such a hard question. And it's very flattering. You're so nice, Julia. Thanks for your question. I think the major finding of that comparative analysis is that the environment is a lot more important in driving life history variation than we ever anticipated. Everyone's always so obsessed about metabolic rate, and I am just sick of it. But I think future studies should try and just like pin down exactly what are the environmental variables that kind of shape life history strategies. So I think species with really widespread distribution, so if there are tropical populations and temperate populations across different continents, I mean, that would be amazing to test some of these ideas. Yeah, does that answer your question?
Yeah, that's great. Thank you, Fonti.
Thank you. Thanks for coming, you guys.
Thanks for that. Do you have any more questions online? Don't forget to put your hand up.
I think we all just need a beer at this point. Oh no, wait, Andrew has a question.
When you showed the, I guess the age course of the distribution of variation, there was a sudden explosion of added genetic variants. You applied at the time of material. What was that about?
So it's actually two things. So I think, well, there's lots of genes switch on and off to promote phenotypic changes associated with sexual maturity. So males in the species tend to have broader heads and they're larger, females have longer bodies. So I think what's captured in that increase in additive genetic variants may be associated with those underlying changes in gene expression. But I also should have said something about how the study was conducted. At about 6 months of age, because the animals, it was just exploding in the lab. There were just so many. Animals were then condensed into communal bins. So they were housed with multiple individuals. And so maybe something to do with that kind of housing condition, whether that's like increased social interactions or competition for basking or food, may have also affected the level of additive genetic variants. I also remembered I didn't repeat that question, but I don't want to.
That's your prerogative.
How we doing, Mike?
I think I haven't seen any more hands go up, so I suspect in terms of online, people have asked questions if they wish to.
Yeah, awesome. Okay.
Yeah.
Well, thanks for coming, guys. There's a lot of online messages too that I should help.
Yeah, there are. And so there were 57 people attending online. So that's a great turnout for you. A testament to the high esteem in which you are held.
Oh, yeah, awesome. Thank you. Thank you, Mike.
The recording will be available for anyone that wants to watch it or that wants to let other people know. It'll go up on Monday. So congratulations, Fonti. I'm going to end the online recording now, and I will come down and say hi in a second.
Sounds good. Thank you, Mike. Catch you later.
Everyone online.